Okay, in this second video, that is the part 2 of reproduction of Oedogonium, I had already explained in the first part, in the first video, on vegetative reproduction, which takes place through fragmentation and achenids, and the asexual reproduction, which takes place through the zoospores, okay, which form in the zoosporangium. Now let us take a look at the sexual reproduction in Oedogonium. So here the sexual reproduction is an advanced oogamous type. Alright, oogamous type are those type of sexual reproduction by which occur due to the union of the male gamete, okay, which is mobile, that is a mobile male, and an immobile female gametes. Okay, so that means the male gamete is mobile, it can move, and whereas the female is immobile. That type of reproduction is referred to as the oogamous type. Now the sexual reproduction in Oedogonium is more common in those forms which grow in stagnant water rather than those which occur in the flowing streams, okay, and it takes place with the help of male and female gametes. Now the male gametes in Oedogonium, they are referred to as the antherozoids. Okay, the antherozoids are produced in the Antheridium, that is the male sexual structure, whereas the female gamete, or also known as the egg, is produced in the oogonium, that is the female reproductive structure. Okay, now both the male and the female gametes, they differ morphologically as well as physiologically, and only one egg is produced in each oogonium, and whereas two antherozoids in each antheridium are formed. That means only one female gamete is produced and two male gametes are formed. Another motile structure, that is the androspore. Remember, this one we will study. The androspore is produced singly in each androsporangium. So you will when we study in detail about the male and the female gametes, you will come to know the difference between these antherozoids and the androspore. Okay, because why? The antherozoids and the eggs, they are the male and the female gamete respectively. Okay, they will reproduce, they will fertilize to give rise to a zygote, which will eventually give rise to a major filament. But here the androspore, you will see, is that spore which is formed in the androsporangium, which will give rise only to the dwarf male. Now let us see what is that dwarf male. Okay, so the deficiency of nitrogen or nitrogenous substances as well as the alkaline pH, these are the important factors, okay, which promote the sexual reproduction in Oedogonium. And in some species, sexual reproduction, it can also be induced. If it's not there, it can be induced in Oedogonium species by placing the major filaments in the atmosphere, which is saturated with carbon dioxide. Now first, before we are going to study about the male and the female gametes, now let us take a look at the distribution of the sex organs. Now species of Oedogonium, they are divided into two groups, okay, on the basis of distribution of sex organs. Now they are divided into the macrandrous species and the nanandrous species. Now what is the difference between these two is that in macrandrous species, the antheridia, that is the male gametes, they are born on filaments of normal size, okay, and species such as Oedogonium nodulosum and Oedogonium fragile, the antheridia as well as the oogonia, the male and the female gametes, they are found on the same filament, Okay, those which are found on the same filament in case of these two species, they are referred to as the macrandrous species, whereas some other Oedogonium species such as Oedogonium crassum and Oedogonium aquaticum, where these Antheridia and the Oedogonia, they are born on different filaments. Okay, so they are born on different filaments, and those species are known as macrandrous dioecious. Dioecious, why? Because the male and the female gametes, they are born on different filaments. Monoecious, because they are on the same filament. Now, although filaments bearing the male and the female sex organs, they are morphologically similar, but they differ physiologically. Now, in case of nanandrous species, just remember that the nanandrous species, they are always dioecious. That means both the Antheridia and the oogonia, they are born on different filaments. Okay, they are not monoecious, but there are some exceptions which we will see later. Now, the male filaments, in case of nanandrous species, the male filaments, they are much smaller as compared to the female filaments, and therefore they are referred to as the dwarf male or the nanandrium. And in these species, that means the filaments bearing the Antheridia and the oogonia, they show morphological distinction. So I hope it's clear that in case of the Oedogonium, okay, they are divided into two groups, the macrandrous and the nanandrous. And in macrandrous, we have monoecious, where they are born on the same filament, and dioecious, where they are born on different filaments. But although they are similar morphologically, but they differ physiologically. Okay. Now, whereas in nanandrous species, they are always dioecious, but why they are called as nanandrous species, just remember that the male filaments here, they are not the same as the male filaments. Okay, they are much more smaller, and they are referred to as the dwarf male, okay, or the nanandrium. So now let us take a look at the male gametes, that is the Antheridium. Okay, let us study about the formation of the male gametes. So here the macrandrous species, okay, in case of macrandrous species we are talking, the development of Antheridium, the macrandrous species form terminal, okay, they form terminal or intercalary Antheridia. Alright, intercalary meaning in between the cells. So they form terminal or intercalary Antheridia, and they form Antheridia by the division of the Antheridium mother cell. So any cell, just remember that any cell, the intercalary cell which has this cap cell, that one, it can act as the Antheridium. Okay, it can act as the Antheridium mother cell. So that is the first thing that you should remember, as in this case in figure A, where this cell, it acts as the Antheridium mother cell, and this cell, it contains a cap cell. Okay, this one we had already studied when we studied the thallus structure. So I hope you still remember. Now, what happened when this acts as the Antheridium mother cell? Now, any cell of the filament with the apical cap here, they can function as Antheridial mother cell, and this cell, it will divide transversely. Okay, it will start dividing transversely, as you can see here in case of figure B and C. Now, it divides transversely into an upper smaller antheridial cell and also a lower larger sister cell. So it divided into two cells, forming the upper smaller antheridial cell and a lower larger sister cell. Okay, so you can see here, this is the sister cell. it is much more larger, and this is the apical cell. Now the latter, this sister cell, it will again undergo division to form a row of 2 to 40. You can see here many cells, around 2 to 40 flat uninucleate antheridia. Okay, the antheridia which has a single nucleus, so they are uninucleate. And also the nucleus, as you can see here, these two, the nucleus, what happened to the sister cell, it will divide into 2 to 40 flat uninucleate antheridia. In this case you can see, whereas in figure D you can see what happened to the nucleus of each antheridium. The nucleus of each antheridium will divide mitotically into 2 nuclei. So each antheridial cell, the nucleus, they will divide mitotically to give rise to 2 nuclei. And as you can see, each nucleus here, it gets surrounded by a cytoplasm. Okay, by some cytoplasm, and it metamorphoses into an antherozoid. Okay, it develop into the antherozoid. These two, after the nuclei, it has divided into 2 daughter nuclei, it metamorphoses, or it develop into the antherozoids. Now thus each antheridium, okay, a single antheridium, it will produces 2 antherozoids. So if many antheridia here are formed, so each antheridia, from each antheridia we will have 2 antherozoids which are formed from these 2 nuclei. Okay, so but in case of there are some species such as the Oedogonium cardiacum. Now in case of that Oedogonium species, the cardiacum, 4 antherozoids are formed. Okay, 4 antherozoids are formed in the antheridium. But in most of the Oedogonium species, just remember that only 2 antherozoids are produced. Now the antherozoid, if you take a look at the antherozoid, it is unicellular, alright, single cell. Then it is uninucleate, it has only a single nucleus. And also you can see these black structures here, they are nothing but they are flagella, and so many flagella are present, so it is multiflagellate structure. Unicellular, uninucleus, and multiflagellate structure. Okay, and if you take a look at the antherozoids here, they are similar to the zoospores. Remember in my previous video where the asexual reproduction it occurs, you can see the zoospores also, it has so many flagella. It is multiflagellate, also it has a single nucleus, and it is also unicellular. So these antherozoids, they are similar to the zoospores. Okay, and also it is smaller in size and has few flagella. It is yellowish in color. Okay, it is yellowish in color due to its reduced plastids. And like zoospores, these antherozoids also, they are liberated by the transverse splitting. Okay, the transverse splitting of the wall of the antheridium. Remember here how the zoospores, they are liberated. It is through the transverse splitting, alright, of the zoosporangium. You can see this zoosporangium here, it split, and thus this vesicle will form this hyaline structure here, it will form, and finally a vesicle surrounded the vesicle here. The zoospore will be form inside this vesicle. So same thing here, it occurs in case of the antherozoids also. Okay, they are released by they are liberated into the outside environment by the transverse splitting of the wall of the antheridium. And you can see the antherozoids, same thing, just like zoospores, they are also enclosed within a hyaline vesicle. okay, within a hyaline vesicle at the time of liberation. Now we will talk about this is in the case of Macrandrus species. Remember, in Macrandrus species, that the antherozoids, they are formed inside the Antheridium, okay, and any cell of the filament which has a cap cell, it can act as the Antheridium mother cell. Now let us talk about the development of the androspore and the dwarf male. Okay, that means this is the case of Nanandrus species. Now in case of Nanandrus species, as I had mentioned earlier, that the Antheridia, alright, the Antheridia, the Antheridia meaning the male sexual structure, it developed in specialized, okay, specialized, then small 2 to 4 cell filament known as the dwarf male, okay, known as the dwarf male. Now how this dwarf male it developed? This dwarf male, it will form from the androspores, okay, from the androspores. These androspores are formed from the androsporangia, alright, they are formed from the androsporangia. Now as you can see the structure of the Oedogonium, as you can see here the structure of Oedogonium, this one we have already studied, alright. So you can see there is no branching, or there is no male, dwarf male in this case. So the dwarf male, it will develop from the androsporangium, okay, the androsporangium, it will start to form and it will produce androspore, and those androspore, they will form the dwarf male. So let us take a look at that one here. As you can see what happened. First, some Nanandrus species like Oedogonium concatenatum, the androsporangia and the Oedogonia, they develop on the same filament, okay. Remember, I'll repeat again, some Nanandrus species, the androsporangia as well as the oogonia, they develop on the same filament, and those are referred to as the gynandrosporus, okay, they are referred to as the gynandrosporus. And there are some Oedogonium species where the two are born on different filaments. You will not find this female, that is the oogonium, and the androsporangia on the same filament like this. The androsporangia, they can form on different filament. So here you can see the oogonium and the androsporangia here, they are on the same filament. So these are referred to as the gynandrosporus, whereas suppose the oogonium, it form in this filament and androsporangia, it develop on another filament, so that will be referred to as the ideoandrosporus, okay, ideoandrosporus. Now the structure of the androsporangia, you can see here the way it developed, okay, it is similar to the development of the the Antheridia in case of Macrandrus, right. The cell will act as the Antheridium mother cell, and it will be divided, giving rise to the upper antheridial cell, small antheridial cell, and a larger sister cell. Again, this one will divide, giving rise to a small antheridial cell and a large sister cell. So it will keep on dividing, and here you can see in this figure where many cells are formed and the nucleus, they start dividing mitotically to give rise to 2 nuclei, and from here it will develop to the antherozoid. So same thing here, if you see the way the androsporangia it form, it develop is similar to the Macrandrus species. Now the structure of androsporangia, it is similar to the Antheridia in Macrandrus, except that in this case, just remember what is the difference. Just take a look at this one. here only a single anthrospore is produced, but here in Macrandrus, two antherozoids are formed. So just remember, two antherozoids are formed, but only a single anthrospore is produced, or else it is similar. Okay, the way the anthrospore and the antherozoids are formed, it is similar. Alright, but just remember, only one anthrospore will be produced in a single sporangia, androsporangia. You can see single nuclei here, which will develop into the single anthrospore. But here you can see the nucleus, it divides mitotically into two nuclei and metamorphoses to give rise to antherozoids, two antherozoids. So here single cell nuclei, and also it will develop into a single anthrospore. Now the nucleus of the androsporangium, okay, the nucleus here of the androsporangium, it does not divide, okay, and the protoplasm, it will directly develop, it will metamorphosis, or it will develop into a single uninucleate multiflagellate androspore. Just remember, this is called the androspore, and it is from this androspore that when it liberated from here, from the androsporangium, also enclosed in a hyaline vesicle, alright, the vesicle will soon dissolve and the androspore, it will swim freely in the water surface. Okay, it will swim in water for some time. Now after some time, what happened, it will be attracted chemotactically. Okay, that means it will attracted chemotactically towards the oogonial mother cell. Suppose there is a cell nearby, there is another filament which has the oogonia, so this will be attracted to this chemotactically, meaning it will move, it will be attracted in response to the influence of a chemical situation. Okay, when there is a chemical stimulation, then it will be attracted to this oogonial mother cell, and it gets attached to it. You can see here, it gets attached to it or any adjacent cell by, you can see it can either gets attached to the mother cell here, the oogonia mother cell, or it gets attached to the adjacent cell here, alright, by its anterior end. Now then here it will start to germinate. So this is the germinating anthrospore. When it germinates, it will give rise to a dwarf male. Okay, this is the dwarf male which developed from the anthrospore. Now the dwarf male, it is similar to any other filament which has this holdfast. Okay, the basal cell, that is the holdfast, and one or more antheridia. You can see antheridial cells here. Now 1, 2, 3, 4. Okay, it has a holdfast and one or more antheridia. Now the nucleus of each antheridium, again they will divide to form mitotically. Okay, they will divide to form two antherozoids, just like in this case. When we have the antheridia, it will give rise to two antherozoids. So same thing in Macrandrus, after the formation of the dwarf male, here the dwarf male, it consists of the basal cell, the holdfast, and the two or more antheridia, and these antheridia will give rise to, each antheridia will give rise to two antherozoids, okay, which is similar in structure to those produced by the Macrandrus species. Alright, so the same thing now, later on we will study this one where the antherozoid, it will come in contact with the oogonial mother cell, it will come in contact with the egg, and fertilization will occur. So this is how the antheridium, it developed in case of Macrandrus and Nanandrus species.